I'm delighted to introduce Dr. Jonathan Spector, who will keynote session chair. Jonathan is and the head of global health here at the Novartis Institutes for Biomedical Research. Jonathan's a trained neonatologist who's led global initiatives in valvular heart disease, sickle cell disease, bringing first world technology and providing access to it in some of the toughest geographies and financially stressed parts of the globe. Jonathan also works shoulder to shoulder, also and always with leaders of the Novartis Institute for Tropical Diseases. Welcome, everybody, and Jonathan, over to you.
Thank you, Jay. Hi, I'm Jonathan Spector, and I work at NITD, as Jay said, the Novartis Institute for Tropical Diseases. And so it's my particular pleasure to introduce Professor Pei-Yong Shi, who is in fact an alumnus of NITD from our days in Singapore, where Pei-Yong directed dengue research. And by the way, an anti-dengue molecule that he nurtured is headed soon to the clinic. We could not imagine a more potent kickoff keynote to this symposium. Pei-Yong Shi is the John Seeley Distinguished Chair in Innovations in Molecular Biology at University of Texas Medical Branch. He's trained in biology at Nanjing Normal University in China, molecular virology at Georgia State, and biochemistry at Yale. Focusing on RNA viruses, drug discovery, and vaccine research, Professor Shi works at the crossroads of academia, industry, and population health, and has made historic discoveries relating to a range of important pathogens with clinical significance. As we'll hear more about in a moment, Professor Shi's basic and translational discoveries were put to work on the front lines of COVID vaccine development and in crucial subsequent surveillance to evaluate SARS-CoV-2 mutant strains. With virtually all of us having ultimately benefited, it's not hyperbole to say that much of the planet owes a debt of gratitude to Professor Shi and his team. So thank you so much for your contributions, Professor, and thank you enormously for being here. To the audience, there will be a Q&A immediately following Pei-Yong's keynote, and you are invited to please submit questions throughout the talk. If you're comfortable to also list your name and institution along with your question, please do so, because we are very excited to hear from you. And with that, welcome again, Pei-Yong, and the floor is yours.
Thank you, Jonathan. Thank you, Jay. And it's really humbling for me to have the opportunity to come back to talk about the coronavirus. As many of you know, I'm not the coronavirologist by training like everybody else. In response to the COVID, everybody feel obligated to work on something. As a virologist, I really feel that's something we should do and to make a difference. That's how we pivoted towards the COVID research. So what I'm going to do today is talk about 4 topics we've contributed to over the last year and a half. I will first talk a little bit about the reverse genetic system. As you know, as a virologist, this is one of the most important tools to study the virus. And then I will talk about some of the reporting virus system that has been utilized to enable small molecular drug discovery, as well as the vaccine development. And then the third point I want to talk about is the emergence of the new variants and their biology in terms of the transmission, and then their implications in the therapeutic antibody therapy, as well as the vaccine efficacy. So first, the reverse genetic system. We developed the first infectious cDNA clone of the SARS-CoV-2. As you know, SARS-CoV-2 contains a very long single-stranded RNA of plus sense polarity. It's about 30 kilobase nucleotide in length. We were one of the first few institutions to receive the clinical viral isolates from CDC colleagues of the first index case viral isolates from Washington State. And then we extracted the RNA from that viral isolate. And because the viral RNA is so long, about 30 kilobase, we used RT-PCR to amplify the cDNA copy through 7 fragments. And then we in vitro ligate those 7 fragments together in the test tube. And then you have a full-length cDNA double-stranded. And then this will be able to encode the viral RNA, and we engineer a T7 promoter at the 5' end of the double-stranded DNA. And using these fused long DNA, we are able to do in vitro transcription and then to synthesize this in vitro RNA, which is recapitulates the viral genome. If we transfect these long RNA into the susceptible cells, we are able to recover virus. And in this way, we are able to manipulate the virus on the petri dish in the laboratory. So on the right shows you the plaques morphology of the recombinant virus from our system, comparing with the clinical isolate. They are identical. The system is very robust to make the virus on the petri dish. So one of the first things we did is we started to engineer reporter genes into the virus. In this case, indicated in the red star, we engineered a luciferase gene into the open reading frame 7 of the viral genome, so we can make the virus. So when this virus infects the cell, it produces the luciferase, and it's an authentic viral infection. So you can see at the bottom, over the time, the luciferase signal is over a million above the background. So it's very robust for drug discovery. So then through collaboration with Gilead, we started to testing the clinically approved antiviral drugs or clinically tested antiviral compounds, as indicated in the next few slides very quickly. So first, we tested the panel of the nucleoside nucleotide analogs. You can see in the list, only remdesivir showed the activity. The second class of compound we looked at is the HIV inhibitors, protease, reverse transcriptase, and integrase inhibitors. Only nafinavir, as indicated here, showed some reasonable in vitro activity. The third class we collaborated on is the hepatitis C compounds. Unfortunately, none of them showed any activity. The fourth category of the compound we tested is the anti-infective drugs. As indicated here, rupintravir, niclosamide, and the chloroquine showed the in vitro cellular activities. So those studies, this assay itself allowed us very quickly to test antiviral activities in the high throughput manner. Along the same line, through collaboration with University of Houston Health Science and the IGM Biosciences, biotechnology company, we started to develop IgM molecule antibody therapy for COVID. And this paper just recently published last week in Nature. So if anybody interested, you can go into details on this. So initially, we used the phage display and identify a very potent IgG antibody we call the COVID-14. On panel A, you can see that the green dots is the epitopes of this the IgG antibody 14, the epitopes. And then we thought that because if we can convert it to the IgM, so that we were thinking about the nasal delivery for the potential treatment. So we re-engineer this IgG into the IgM format. And then once we re-engineer that, we saw that the potency, as indicated in panel E, that increases by hundreds folds. And then more importantly, the IgM format of this antibody can cross inhibiting the different new variants, as indicated at panel D, the B117, which is the UK variant, the P1, which is the Brazil variant, and then the B1351, which is the South African variant. You can see IgM across showed very potent activities. And then in contrast, the IgG format, it decreases its potency in vitro. And also, we show this IgM through nasal delivery in the mouse model. It is very, very potent, as indicated in panel F, which is the prophylactic treatment 6 hours before the infection in the animal model. You can see the dose response of inhibition. And then on the panel G is the inhibition for therapeutics. Which is 6 hours post-infection. So these data showed the promise of using the IgM formality for potential not only for COVID, but for future respiratory disease infection. And then besides the luciferase system we developed, we also engineered the fluorescent genes into the virus, in this case, the M neon green, and we were able to generate the M neon green. So when the virus infects the cell, it turns the cell green. This is very useful for high throughput measuring of the neutralizing antibodies, which is very, very important for the vaccine and the plasma therapy early in the last year. So the assay is very simple. 96-well format, you precede the cells, uninfected cells on day 0, and then the next day you mix your antibody with your fluorescent viruses and then infect the cells for 16 hours, and then you're able to use the high content imaging to calculate the neutralizing antibody activities. So using this platform, we worked very closely together with Pfizer to enable their first vaccine neutralizing antibody data for the phase 1, phase 2, and the phase 3, even the preclinical development. As indicated here in these slides, the dose response of 10 microgram, 30 microgram, and 100 microgram, and using these assay to measure after the dosing, the immune response, the neutralizing activity. And then this assay was also used by Moderna to look at their neutralizing activities. In this slide specifically showing the durability of the human vaccinated by the vaccine 9 months over time, the durability of the neutralizing activities as shown here for 3 age groups. So you can see there is a decay of the neutralizing activity, but even after 6 months, there is still substantial neutralizing activity retained in those individuals. So although these authentic virus containing the reporter gene is very, very useful, one of the limitation, I would say the major limitation of the system is it requires biosafety level 3 because SARS-CoV-2 is the BSL-3 agent. So we started to think whether we can engineer the system that we can do the assay or manipulate the virus, authentic virus, at the BSL-2 facility. So this slide summarizes one of our recent work that targeted towards this goal. So we designed a so-called Transcomplementation system, which have been widely used in many virus systems. So what we did is we deleted 2 genes of the viral genome. In this case, open reading frame 3 and the envelope gene. And then we also engineer the reporter gene, in this case, M neon green, into the system, into the viral genome. And then we made a transcomplementing cell line that expresses those 2 missing genes. So as you can see, if you electroporate or transfect the genome with the 2 missing genes into the cells, it's going to transcomplement and make the viruses. And this virus is just like authentic virus, except the genome inside the particle missed 2 genes. And then this, if you infect the normal cells with these viruses, you will be able to authentically infect and enter and then replicate inside the newly infected cells. But because they miss those 2 genes for the packaging, so they'll not be able to make the next round of progeny viruses. So this is enable us to do the assay in a single run. So using these assays, we tested in vitro, in vivo, by consulting with the institutional biosafety committees, with addressing all the experiments they require to do. At least UTMB has already approved for using this system at the BSL-2 level, and we are in the final stage consulting with NIH to downgrade this system to the BSL-2. So I think this could be a game change for many of the labs who don't have the BSL-3 facilities. And we've also tested in this paper that this system can be very robustly used for measuring the neutralizing antibodies, antiviral activities, and etc. So now I'm going to switch gear to talk about the variants. The first dominant mutations after the pandemic is this so-called the spike D614 mutation. So as we know, the initial sequence when it's published, the virus at this position is aspartic acid at D614. And then at the late February of last year, there is a mutation started to be identified, which changes this residue to a glycine. So as you can see from the top, as time goes on, the population more shifted towards the G residue. And then the question is, what does this mean? Does it increases viral fitnesses, the transmission, as well as changes the neutralizing activities of the antibodies or the vaccine? So we're very interested in these questions. So what we did is we used the reverse genetic system and made a pair of the isogenic viruses, everything identical, except that this residue has been changed from D to G. As you can see in panel B, the plaque morphology are identical between the two viruses. And then initially, we used the CALO3 cell line, which is the human lung epithelial cells, to look at the growth kinetics over the time, as indicated in panel C. You can see the black means the Y type D614, the red means G614. You can see there is a slight advantage in terms of replication, amplification for the mutant virus. And then the next experiment we did is we used the hamster model to compare the pathogenesis and the viral load of the viruses upon intranasal infection. So as indicated here in panel B, again, the black means the Y type, the red indicate the mutant, and you can see the weight loss in panel B, and they all cause the weight loss to an equal level. So the mutation does not seem to change the virulence of the viruses in the animal model. And then when we measure the viral load at the respiratory tract of the infected animals, as indicated in panel C, you can see on day 4, the nasal wash, there are more than 10-fold increase for the mutant viruses. These are the infectious viruses, as well as the trachea. But in contrast, in the lung, there is no significant differences between the two viruses. So these data clearly demonstrate that this mutation does not necessarily enhance the pathogenesis of the virus, but that significantly improves its viral replication fitnesses of the virus, particularly in the upper respiratory tract. And that indicates that this might enhance the transmission of the viruses by this mutation. So to further confirm these results, the animal results, we use the human primary airway culture as indicated here. In panel B, we're doing the growth kinetics of the two viruses on the human airway culture. So you can see the mutant virus in red replicates more efficiently than the original virus. And then we also, in panel C and D, we're doing competition experiments to further validate these results. So what we did in panel C is we mixed the 1-to-1 ratio of the Y type and the mutant virus. And then over the time, we measure the progeny virus, the population ratio between the mutant viruses versus original Y type virus. You can see over the 5 days, the mutant virus wins by over 10-fold indicated here. In panel D, what we did is we changed the input virus from 9 to 1. 9 for portion of the Y type virus, only 1 portion of the mutant virus. As you can see, again, over the time, the mutant virus wins the competition. So these data clearly demonstrate that this mutation enhances the viral replication of the virus on the human airway primary culture. So then we also looked at the stability of the viruses caused by this mutation. What we did is we incubate the equal amount of infectious viruses, mutant Y type, at different temperatures, 42, 37, panel B, and at the bottom C, panel C, 33 degree, which mimics the up airway temperature. So you can see the red viruses sustained more infectivity over the time. So that indicates that the mutation may enhance the thermal stability of the viruses, which might contribute to the high replication levels in the respiratory tract. So after we published our data, and two other groups, as indicated here, they directly used the animal transmission model, which is the hamster and the ferrets model, to directly demonstrate that this mutation can enhance the transmission directly between the animals. So in terms of mechanism, how these D614 enhances the infectivity or the replication fitnesses, the current explanation based on the structure is, as we know, the spike is a trimer and it has the receptor binding domain, which is critical for binding to the receptor human S2. So the mutation triggers or promotes the receptor binding domain, as indicated in the bottom of the pictures, into an up conformation, which is a conformation required to engage the human S2 binding, to engage the viral infection. So this is the potential mechanisms of why these, at least one of the potential mechanisms of D614 that enhances the viral replication. So now I'm going to switch gear to talk about another variant. The reason I talk about this is because this B117 variant originated from UK, initially identified, and this is, in terms of prevalence in the US, it's over 65 to 70% prevalence among the circulating strains. So that's why we are interested in studying them. So what we did is we constructed in panel A a panel of mutant viruses. We're interested in asking whether the spike mutations in the B117 variant increases the transmission. That's question number one. Number two, if so, what mutations in the spike B117 that plays a major role in determining the enhanced replication or transmission? So in panel A, we made 8 individual mutant viruses. Each mutant contains a single mutation from the B117 spike, and there are total 8 mutations in the spike from this B117. And then panel B shows the locations of these residues, mutations, and panel C shows the viruses we can recover that. So to screen which mutations among those 8 contributes or have any role in terms of viral replication fitnesses, we use the hamster model, in this case using the transmission model. So what we did is we inoculate intranasally the hamsters, and then we measure the first inoculated animals, their RNA levels in the respiratory tract, and also on day 1 post-infection of this animal, we put a clean animal into the same cage for 8 hours to allow transmission to occur. And then after 8 hours of co-housing, we separate these 2 animals, and then we monitor both the donor animal as well as the recipient, the animal, their respiratory tract viral loading. And then when we do this experiment, we do also use the competition experiment. So we mix 1-to-1 each individual mutant versus the Y type 1-to-1 to the inoculation. And then we assay the ratio between the mutant versus the Y type. So the number, any number above 1 indicates the mutant has advantages. So you can see panel B, which is the nasal wash of the donor animals on day 4 post-inoculation. So you can see the individual mutant I indicated at the bottom. You can see that 501 mutation, M501, increases the viral replication for the mutant. To a small extent, the delta 69, 70 also has slight increase. And we also made the 8X, which contains all the mutations of the spike. We call the 8X UK. You can see that it also has advantages. And then for the recipient, the nasal wash, the same pattern, the 501 and 8X, and to a less extent, 69, 70 deletion has advantages. So once we know this 501 is important for this enhanced replication and the transmission, and then we characterize this 501 mutant as indicated here. So what we did is, again, 1-to-1 ratio, the 501 versus the wild type virus, and then we assay in panel A the donor nasal wash. You can see over 4 days, the mutant virus has twofold advantages in the replication. In panel B is the nasal wash of the recipient animals. Again, consistently shows advantage. And the other organs, which also, I'm not going to go into the details, they also showed advantages as indicated by the fold of increase. So then to validate these hamster model, we also use the human airway primary culture as indicated here in panel A on the right. You can see the increase of the ratio of the 501 mutant viruses over 1 to 5 days, 2 to 4-fold, 5-fold increase in terms of replication efficiency. And then we also look at the mechanism of this enhancement of 501 mutation. We purify the protein, and then we performed by physical interaction to look at the KD, K on, K off, and that clearly shows that there is over 100-fold improvement in binding to human S2 once you have this 501 mutation. So we think that might explain the mechanism. Through 501 mutation, it significantly increases the binding affinity to the receptors and therefore enhance the viral replication. So now in the last few minutes, I'm going to talk about other variants and their effect on the neutralizing antibody as well as the potential vaccine efficacy. So we've been working very closely with Pfizer and the BioNTech to systematically to follow on what are the new variants become more dominant in the different geographic regions. And then we're very interested in looking at whether those mutations, particularly in the spike gene, how they affect the potential neutralization of the individuals who have been vaccinated. So our approach is distinct from the other groups. So we use the reverse genetic system to make chimeric virus. Those viruses are authentic viruses. So what we do is we use the Washington isolates as indicated here at on the left, the top. Those are the original virus, the Washington isolates. And then what we do is we swap the complete spike gene with the different new variant indicated here, B117, which is a UK variant, and then B1351, which is the South African variant, and then P1, which is a Brazil variant. So only the difference is the spike on the surface. And then through collaboration with Pfizer and the BioNTech, we took the same 20 human serum from individuals who have been fully vaccinated by 2 doses, as indicated on the right by this sample collection or vaccination scheme. So day 1, day 21, 2 doses, and then those samples were collected 2 weeks and 4 weeks post the vaccination. And then these slides summarizes a couple of month results. So you can see what we do is, because we made all the panel of the chimeric spike viruses, and then we use the same 20 patient serum, and we measure for each of those serum against each of those viruses, using the plaque reduction neutralizing assay, which is the gold standard in the serology assay to measure neutralizing activity. So you can see at the bottom, let's look at the bottom first. So on the bottom, on the left, we have the Washington, the Y-type isolates. And then we have a B117, the UK, the P1 is the Brazil, and the B1351 is the South Africa. You can see the dramatic drop by almost threefold in terms of neutralizing activity. The South African variant indicated in red box compared with the original, the original U.S. Washington isolates. So this South African variant represents the most concern among all the variants so far in terms of its neutralizing reduction activity. And then also on the right at the bottom, we've tested the B1427, 429, which is the California variant, the B1526, which is the New York variant, and then also the B117 plus 484K. The 484K, E484K is the mutation which is dominant appeared in the variants, and that confirmed the resistance to many therapeutic antibodies, even those approved by FDA for EUA. So that's why, and then this mutation also occurred in the UK backbone. So that's why we tested there. It doesn't seem to significantly reduce the neutralizing activities against the vaccinated human serum. And then at the top on the right, this is the new data, and it's just accepted in Nature last week. So these are the new variants we tested. Among them, the India variants, as we heard a lot, and Indian, there were several sublineages indicated here, and we found the B1 indicated in the red box, the B1617.1 spike confirms the most dramatic reduction, threefold reduction in terms of the neutralizing activity. So to sum it up, all the current data based on our system that the two variants is of concern. One is the South Africa variant, and then the other is the the Indian version 1 of the variants. The other version did not seem to dramatically reduce the neutralizing activity. And then finally, I really want to thank our collaborators and the people in the lab. As I mentioned, our vaccine is through close collaboration with colleagues Pfizer, BioNTech, and then the antivirus project, we've been closely working with Gilead colleagues. And then the IgM antibody, we've been working with the UT Health Houston, and then also the IgM Bioscience, the biotech company, and other collaborators, Washington University of Mike Diamond and colleagues at UTMB. And I also want to thank, going back to the NIBR postdoc program, and Dr. Pei-Yong Shi, as indicated here, the first on the top left, and he is the recipient of the NIBR postdoc when I was still at the NITD. And we were so fortunate that the NIBR supported him even after he moved to Texas with me. And he is really the person who behind pretty much everything I talked about, making the first reverse genetic system and working on different fronts as I presented today. I'm really thankful for the NIBR postdoc program too. So with this, I will stop here and look forward discussion with all of you. Thank you for your attention.
Pei-Yong, thank you. Thank you so much. That is a privilege to hear your personal account of this science with such breathtaking clinical significance. The questions are rolling in, and so we're going to go directly to them. The first one is coming from our very own Novartis infectious disease specialist, Variance Seagull, who's helping out behind the scenes also with this talk. And the question is, can the enhanced viral replication in the upper respiratory tract observed experimentally with the D614G variant result in increased disease severity indirectly through an immune system imbalance?
Yeah, we still so busy with some of the primary tasks, and obviously this is a very, very interesting question. And over the past year and a half, we're very much just focusing on the virus side, very obvious things to monitor. But I do think the immune response, because of the differences of the replication capacities or levels in different regions of the respiratory tract, will contribute to the different immune response, which will really contribute to the disease outcome. And also, I think the field has been very much focusing on the spike right now. And there are other mutations outside the spike that really needs to be also looked at. What do they do, right? And do they really also, through modulating the immune response, or as well as modulating the viral replication? So it's an open question. There is a lot to do here.
Perfect. Thank you so much. And actually, on that question, the spike, can you comment a bit more on our ability to predict the likelihood of mutations outside the spike protein that are likely to emerge or that may emerge under viral pressure?
Yeah, I think, there are different systems has been used. For example, people are using the yeast system to mutate pretty much every single amino acid to the different options to look at the bindings, and then you display them, and then you look at the enhanced binding of each mutations, and then you come back to look at them using most of them using the pseudotype viruses to look at how they, in fact, if you engineer those identified mutations from the yeast to the pseudotype viruses, VSV or lentiviruses, and then you look at their function. So there is a lot of this kind of studies ongoing. So theoretically, that is able to allow us to identify certain potential mutations. But of course, as a virologist, we also potentially you can do through selections, right? But of course, with the current gain of function and all those studies, we have to be very, very careful of what can be done and what is worth doing. So this is something of a topic of ethics and other things needs to be collectively discussed. There is a lot of things we can do, can think about. Absolutely. Important question.
Thank you for that. Another one coming in from Neil Hunter. Could you please comment on the suitability of the hamster model, since there are only low levels of ACE2 expression?
A good question, and there is different models over the time have been developed. Hamster, right now, I think my personal opinion is among all the models, that might be the one of the best model, because if you look at the mouse model, there are a human S2 expressing transgenic mice, or you can deliver it using other viral vectors to express the human S2 into the mouse to make them susceptible. I think that is obviously a reasonable model, but those models, the virus goes to the brain and causes neurological disease, etc. So every model has its advantages and disadvantages. And then the monkey model, the non-human primate model, it doesn't seem to have a very significant disease. They will develop viral replication, certain level of disease, but not as significant as we wish for. But the hamsters, they develop, as I showed, develop the significant, very clear weight loss and pathogenesis. And then you can also do transmission in that. It's very well established by many, many labs around the world. And then also for transmission, there is ferrets model, but ferret model is very good for transmission. But in terms of disease, ferrets, the disease after the infection are very minor. So, among all, personally, I still feel hamster is a good model to look at this for both, vaccine and immune response. The only shortcoming of the hamster is the reagents. The antibodies against the hamsters and all those has some limitations.
Perfect, thank you. New question coming from Wes Van Voorhis from University of Washington, infectious diseases. Pei-Yong, do you think a threefold reduction in neutralization will lead to breakthrough infection?
That's a very good question. I think we are only, most of our energy and effort right now is focusing on the neutralizing antibodies. As we know, the immune protective mechanisms is more than just the neutralizing antibodies. Although neutralizing antibody might be one of the major ones, there are other protective mechanisms through T cells, through non-neutralizing antibody, cytotoxicity, complement, and etc. So those are still coming along. There are a lot of great studies coming out, but they're just not as easy to measure very quickly. So that's why a lot of studies on the neutralizing antibody activities, but neutralizing antibody will never be able to replace the other mechanisms, as well, more importantly, the real world observation of the vaccine efficacy. So the threefold, fourfold reduction in our study against a certain variants, but for example, the South Africa, the Indian variants, and there are recent studies from the real world vaccinated individuals, the efficiency, although the N numbers is still not ideal, but you can see even for the South Africa variants, they are against the Pfizer's vaccine, it's reported over 75% efficacious in those real world data, as well as the recent Indian variants in UK study showed they are close to the 88% efficacy. So there is still a lot to be looked at closely, but I think neutralizing antibody is not the only answer to that, but might be a very important one. So I think really in the end, it really needs to be the real world data to look at the breakthroughs.
Thank you. Question coming in from Dario Bichara, who's dialing in from Argentina. Could the enhanced proliferation in the upper respiratory tract mean that a vaccinated patient can still be a vector?
I don't know on the vaccinated enhancement. There are recently, there is one paper, and I think, particularly coming from the dengue field, the enhancement has been really in the field for a long time. It is true that there is a different serotype enhancement through the antibody-mediated effect. And I think there is a lot to still remains to be carefully looked at. But I think as the vaccine developers are closely, and the whole scientific field is closely monitoring the situation. It's an important one, but we need to closely monitor that.
Perfect, thank you. Pei-Yong, we're just about at time here. We have, but we'll try to squeeze in one or two more questions. A few questions coming in asking about, did you see new mutations generated in the animal models?
We haven't really got time to look at these, but for some of the treatment studies, for example, the IgM treated animals, in those animal models, we did sequence some of those, but not systematically. We don't see short time any mutations. But remember, the 501 mutation, the 501, M501 mutation Y, initially, this mutation was reported by the Chinese scientists in the mouse-adapted model. And this is why a long time ago, our lab already started to engineer. The day I saw the Chinese scientists report on mutating this 501 through the mouse-adapted isolate, and we engineered that because this will allow us to build up a strain that can infect the mice and then do all the antiviral and the other studies. So there is a convergence, from both mouse adaptation and the human, like a 501 is independently evolved in the different geographic locations like UK, Brazil, and the South Africa variants. They all independently to converge to have that mutation, while there are some other mutations. So we haven't really looked at the mouse adapted or other animal adapted variants. yet.
Pei-Yong, I'm sure we could keep going. There's still lots of questions, but we're going to have to give some of the other speakers time to contribute as well. Thank you so much. We could not have imagined a more compelling way to kick off this day of COVID-focused science. So thank you again, Pei-Yong, for being with us. And
thank you, Jonathan, for having me.
It's my pleasure. Thank you so much.